Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, a very warm welcome again to the biology learning lesson. As usual, I'm teacher Herbert, and I will be taking you through the biology of Year 3. And the lesson of today is concerning about vesicle transport and the summary of all active transport. I hope everybody remember that we talked about passive transport, where we talked about osmosis, where we talked about diffusion, and where we talked about facilitated diffusion. And we said those processes do not require energy. There is no need of energy. They are passive processes. Students, if I talk about passive processes, they move without using the energy. And now we're going to talk about active transport. And we talked about active transport, where we talked about movement of molecules. Particularly, I'm talking about carrier proteins, where they can move from one membrane to another membrane by the help of energy. And I mentioned the energy that is going to be moving is in form of ATP. And student, I think you do remember what is ATP in full. Student, I mentioned ATP in full is adenosine triphosphate. We get adenosine triphosphate, that is 3-phosphate group, and we mentioned that the third phosphate group is more energy rich. So let's go to what the lesson of today, vesicle transport, and a summary of active transport as I mentioned it earlier. So I'm teacher Herbert. Let's go through the lesson. The running objective of that lesson of today is to make a review of what you call vesicle transport. And it is explaining the factors that affect active transport. I hope everybody remember what are the factors that can affect active transport. The second learning objective is to explain vesicle transport. Here it means we are going to go through the endocytosis process, and we're going to go through exocytosis process. So let's start our lesson of the vesicle transport. And I mentioned that vesicle transport involves two processes, endocytosis and exocytosis. What are vesicle transport? Vesicle transport, or other bodies in the cytoplasm, moves micromolecules. If you talk about micromolecules, I mean large molecules, not small molecules. Small molecules are mostly carried by the passive process. Passive process carries large molecules, what you call small molecules. But when it comes to active transport, it carries macromolecules. Talk about macromolecules, it means large molecules or large particles across the plasma membrane. And that plasma membrane must be able to be partially permeable, must be able to allow some molecules to pass through. I think, student, you do remember what is a semi-permeable membrane. What is a semi-permeable membrane? And what is a selectively permeable membrane? Semi-permeable, it means some molecules can pass through. Partially permeable, it means that it can allow some molecules, let me say glucose to pass through, but it limits other molecules to pass through. Not all molecules can pass through. So because they are just too large to pass through the plasma membrane or to move through the transport, or through a transport protein. Because they are very large, they don't pass through the plasma membrane. If they don't pass there, what happens? It needs energy to move those proteins, to move from one membrane to another membrane, because those molecules, they are very important. Why are they important? The body needs them. The cells need them. Take an example. Some say they need glucose. You remember I talked about what are the materials that must be supplied to a cell if to survive. Take an example like glucose. Take an example like proteins. They have to move from one membrane to another membrane in order to help the cell survive. So let's go to There are two types of vesicle transport, as I mentioned. We have endocytosis and exocytosis. Student, let's try to have the difference between under endocytosis, student, under endocytosis. We have two types. We have phagocytosis, and we also have pinocytosis, and we also have exocytosis. Let's try to differentiate the three: the phagocytosis, the exocytosis, and the pinocytosis. Let's go through our lesson. How important are these processes in a cell? Take an example, how endocytosis is very important to a cell. As I mentioned earlier, the endocytosis and exocytosis, these are called vesicle transports. They transport large molecules. And these large molecules, they are needed by a cell if a cell is to survive. Let's talk about the importance of endocytosis. It's the process of capturing substances or particles from outside the cell by engulfing. If you talk about engulfing, that is surrounding something, and you take it from outside to inside. You get what I'm talking about, student? Meaning you take substance from outside, particles from outside, and then you take them inside the body from the surrounding environment. And the membrane folds, if I talk about folding, that is, let me just have some diagram. I have substance, this is a particle. Like this can call it be a particle. It can just be a food particle. It can be a food particle. That food particle can be surrounded. Surrounded, you know how amoeba feeds. After surrounding, now it is going to be taken from outside to inside. That one involves what you call folding the membrane, folding the plasma membrane. Folding of the membrane over the substance, and it becomes completely enclosed by the membrane. After surrounding it, now it is going to be engulfed. This is the membrane. After surrounding it now, as you can see, we have something has been surrounded and engulfed. And after being surrounded and engulfed, now it is going to be taken from outside to inside. And that is what we call endocytosis. As I mentioned earlier, student, as I mentioned earlier, there are two types of endocytosis. Number one is phagocytosis. Number two is pinocytosis. Who can give me an example of phagocytosis? I think you do remember how some of the organisms feed. Let me talk about bacteria. Let me talk about amoeba. Let me talk about how the white blood cells, how they normally engulf some of the microbes and take them from outside to inside. And after getting them, they destroy these bacteria, which can cause disease. Let me have amoeba. You know, amoeba has no a definite type. It has some structures, which is called pseudopodia. If you remember, there's what you call pseudopodia. Pseudopodia. Pseudopodia are armed structures. They are armed structures that are found in amoeba, that are used by amoeba to feed. How amoeba feeds, engulfing and surrounding. That one is called phagocytosis. Then we have another which is called pinocytosis. Pinocytosis is a type of endocytosis that involves taking liquid substances from outside into inside. If I talk about liquid substance, I mean fluids. Take an example like extracellular fluids. They are needed by the cell. They need to be taken from outside to inside. So that one is called pinocytosis. Let's have the definitions. Let's go the definition. Phagocytosis. As I mentioned earlier, that one is called cellular eating. When you are eating, you can eat solid substances, not liquid substances. So phagocytosis can also be referred to as a cellular eating. It occurs when the dissolved materials enters the cell. The plasma membrane engulfs the solid materials from forming what we call phagocytic vesicles. phagocytic vesicles, that is some of the substances from outside to inside. This is mainly observed in white blood cells, as I mentioned. They are found in white blood cells. If I say white blood cells, this is commonly found in white blood cells. You know what are white blood cells? White blood cells, that is WBC. WBC. It is commonly found in white blood cells, where the white blood cell has to engulf and surround the soiled part, the microbes, and after surrounding it, you are now getting healed from the disease. Some of the microbes can enter into our body. But student, you remember that we have lines of defense. We have the skin, which is acting as a line of defense, but also the white blood cells also acts as a line of defense of engulfing and surrounding microbes, and after surrounding, they destroy them, and after destroying them with the enzymes, and now you become okay from the disease. Let's go to the diagram that shows the process of surrounding and engulfing, surrounding soiled particles. Here I have a very good diagrams. The diagrams is showing the structure of a white blood cell. These are the structure, these are the bacteria. They are outside. They need to be taken from outside to inside. Are you getting me, students? Student, I think you are seeing the diagram. We have how many diagrams? We have almost 4 diagrams. This is diagram 1, diagram 2, diagram 3, diagram 4. Now you see white blood cells is the phagocyte. Is surrounding the bacteria. And after surrounding, we have pseudopodia. As I mentioned, these are armed structures which are responsible to surround using the plasma membrane. They surround the bacteria, and after surrounding them, they digest, they break down these soiled particles, and after breaking them down, now they're going to be released, the bacteria, the microbial products are going to be released as a waste product from the cell. Let's have another diagram that is trying to have how to talk about pinocytosis. Pinocytosis refers to cellular drinking or cell drinking. You know how to drink water. So taking substance, when you are drinking water, you take it from the cup, you take into your body. That one is referred as cellular drinking. That one is called pinocytosis. I said it occurs when the plasma membrane folds inwards to form a channel around the dissolved substance to enter the cell. These dissolved substances refers to the fluid, liquid. Take them from outside into the inside. That one is pinocytosis, is referred to as cell drinking. Good. Let's go to the diagram. The diagram that tries to illustrate the process of pinocytosis. I have a diagram. We have extracellular fluid. It is outside of the cell, not inside. Now that these substances that are in liquid form, they are going to be taken from outside to inside. There's a folding. You see, there's something like a fold. Folding around these substances. Now, after folding them, they take them from, after folding, you see, this is the first step, this is the second step. And after folding its membrane, now the substance, the extracellular fluid, is taken from outside to inside in the cytoplasm. This is the membrane folds, and after making that one, I mentioned about what you call folding of the membrane. And the substance is going to be taken from outside to inside. And that one is referred to as pinocytosis. Simply, it refers to cell drinking, taking liquid substance from outside to inside. Let's go to simple I talked about. Occurs, I said this one, folding in, this is supposed to be forming the channels, and around this one, the substance enters into the cell. Let's go to the diagram of endocytosis. Simple summarizing the diagram of endocytosis. This is endocytosis. We have some substances, they are here. They are substances which are outside. They are going to be taken from, the membrane is going to be folding. You can see the membrane folds around the substances, and after folding, they are going to be taken from outside to inside. Let's talk about this one. That one is trying to summarize what you call endocytosis. Taking substances from outside to inside. We have exocytosis. Exocytosis, I would try to remind you, Our body has waste products. They are found inside. There are some substances which are found inside. They need to be taken outside. That one is done by exocytosis. The word exo, it is removing substances which are inside to the outside, simply. We have the diagram. This one is called a vesicle membrane. It's going to be surrounding it. After surrounding, this one fuses with the vesicle membrane, and now the substance is going to be released out from the cell to the outside of the cell. That one is referred to as exocytosis. Let's talk about some of the factors. What are the factors? As students, we are making a summary of the lesson. What are the factors that can affect active transport? As I mentioned earlier, active transport requires energy. It is energy, it uses energy. If I talk about energy, it means it is energy use. It uses the energy. So simply, if it uses the energy, it means there must be some of the factors that can affect the active transport. A number of factors that can affect active transport: we have concentration of oxygen, we have concentration of glucose, we have temperature, we have enzyme inhibitors, and so on and so forth. Let's go through these factors. Let's go through these factors and let's talk about how I said factors that can affect active transport. Concentration of oxygen. As you know, if there is more concentration of oxygen, more energy is going to be produced. If there is less concentration of oxygen, less energy is going to be produced. I think all of you do remember, when you are doing sports, you are using more oxygen. And if you use, the muscles need more oxygen. That's why most of the time you breathe taking more oxygen that we're going to be used in a cellular respiration. So I said high concentration of oxygen, the cell produces more energy. You get me very well? Produces more energy. Less concentration of oxygen, less energy is going to be produced. This leads to the rapid active transport by the cell protein carriers. Number 2, glucose. Student, I think you know the importance of glucose. When you break down glucose, more energy is going to be produced. And we normally say that energy is going to be in the form of ATP. So more concentration of oxygen can produce more energy. Less concentration of glucose, less energy is going to be produced. Let us read what is written there. The amount of energy produced by the cell depends on amount of glucose present or amount of glucose available. When more glucose is available, the cell produces more energy. This leads to what you call a rapid active transport. I think you are following me very well. We have the second one is temperature. I think you do remember what is temperature. Our body temperature is ranging from 37 degrees Celsius. When the temperature is above, it affects body metabolism. It affects enzymes. And you do remember, enzymes are protein in nature. High temperature denatures enzymes. Low temperature makes an enzyme to become inactive. So let's talk about how is active transport is going to be affected by high temperature. High temperature can destroy or denature protein carriers. As I told you, our body is made of proteins. So these protein carriers that are taking substances from one cell to another cell can be affected by high temperature in the cell. This stops the active transport. Active transport works best under a normal temperature. In human, it means that the normal temperature is 37 degrees Celsius. When it is below that, it means that these protein carriers is going to be destroyed and it will affect active transport. We have enzyme inhibitors. What are enzyme inhibitors? These are substances that can affect or can inhibit the enzyme's activities. And we know there are very many enzymes that participate in many activities of active transport. Enzyme is a substance which speeds up. It can speed up or it can slow down the rate of reaction taking place. It means that we need just enzymes inhibitors are chemical substances which are poisonous or make the enzyme inactive. If enzymes involved in releasing of energy, we said energy production, talk about active transport, energy production. If there are some enzymes that are responsible for participating in that participates in releasing of energy and are going to be affected by high temperature, eventually that one will lead into a release production of energy. So that involves in release of energy during respiration are affected. There will be a lack of energy, and this will prevent active transport from taking place. If there is less energy, that one affects active transport. We have what you call pH. A pH. Student, when you talk about pH, most of you don't understand what you call pH, the word pH. Let me just try maybe to use a colored chalk. pH. pH. pH is the degree of acidity and alkalinity. We know that some of the enzymes work best in acidic environment. Some, they work best in alkaline environment. And how are these active transport are going to be affected by pH? Abnormal change in pH, when there is a change in pH, a slight change in pH can affect the active transport. I think you are getting me very well. Abnormal change in pH in the environment of the cell can irritate. If I talk about irritate, can destroy the structures of protein carriers. So once you change the structure of carrier proteins, because of the change of the pH, that one will affect the active transport. This will lead to reduce the ability to transport molecules across the membrane. Once there is a slight change of pH across the membrane, eventually that one will affect the active transport. You get me very well? Can I repeat the statement? I talked about the pH. The pH, the abnormal change of pH in the environment of a cell, you talk about the surrounding environment of the cell, will irritate or will destroy the structure of protein carriers. And if the structures of protein carriers are destroyed, automatically most of these protein carriers, they have specific structures which enable them to carry some molecules from one cell to another cell. So this will reduce the ability to transport molecules if their structures has been destroyed, if their structures has been irritated and across the cell membrane. So that one will also affect the cell structure, the active transport. Let's go through the assessment to the lesson. Student, I think we've been hearing active transport. We talked about endocytosis. We talked about, who can remind me about endocytosis? Student, I said endocytosis has two types, the phagocytosis and phagocytosis and pinocytosis. And now phagocytosis, I mentioned it is that one involving surrounding engulfing. I talked about cell eating. Do you remember? Cell eating, phagocytosis. We gave an example of phagocytosis, amoeba, white blood cells. Then we talked about pinocytosis, cell drinking of fluid substances from outside to inside the cell, because the cell needs those fluids in order to survive. And we talked about exocytosis, removing substances from inside the cell to the outside of the cell. Let me give an example. Waste product in our body has to be removed because they can affect our body metabolism. You get me very well? So removing of waste substances from our body is done by exocytosis. Not only that, take an example, the reabsorption of glucose by the kidney is also done by exocytosis. Let's talk about the assignment. Take your exercise books. I remind you to take your exercise books and do the assignment. White blood cells ingest solid particles by the following vesicles. Describe which process. You describe the process. What is that process? How does amoeba takes these solid particles? I can do it with you. I will do one for you. The white blood cells ingest the solid particles by forming vesicles. This describes which process. The process is referred to, that is question 1. The answer of question 1 is phagocytosis. So I'm giving you the answer of this process, and where necessary, you have to draw the diagram. So the now question number 2, give these characteristics. Given these characteristics, we have a number of characteristics. The characteristics that we have requires energy. Which one that requires energy? Requires carrier proteins, requires membrane channels, and requires vesicles. So it means that choose the characteristics that defines the exocytosis. I hope you have the answer. The answer for me, I have 1 is endocytosis, then 2 you get Roman 1 and Roman 2, and even Roman 5, that one requires energy. Let's go to the summary now. I'm making a summary of what we've been talking about. Let me just make a summary. Active transport, I'm making a summary of the lesson. The summary of the lesson is here. Active transport involves the use of energy. I think I mentioned it. During active transport, molecules move against concentration gradient. They don't move down the concentration gradient. They are moving from lower to higher concentration. It is down the concentration gradient. I'm making a summary of the lesson. And factors that affect active transport includes concentration of oxygen, glucose, temperature, enzyme inhibitors, and pH. We are rewinding our lesson of active transport, and we are rewinding our lesson. Quickly, let's leave the homework. I said talk about how is the concentration of oxygen affect active transport, and you explain it why, and identify the location in plants and animal tissues where active transport takes place. So, student, thank you very much for watching and listening, and thank you very much for watching and listening, and stay home and wash your hands with clean water and soap. May God bless you.